Hello, and welcome to another edition of Inside the Lab with the CMBC. I'm Dietrich Stout, the Associate Director of the Center, and today we are being joined by Dr. Aubrey Kelly. Dr. Kelly is one of those interdisciplinary researchers that it's kind of hard to put a specific label on, but broadly speaking, she studies the neural, behavioral, environmental, and evolutionary foundations of social behavior in animals, ranging from birds to rodents. She completed her PhD in biology and neuroscience at Indiana University, studying the neural mechanisms of social behavior in finches. She then did a postdoc at Cornell, where she studied the development in socially monogamous prairie voles. She joined the Emory Department of Psychology in 2018, and her lab is now working on various projects with prairie voles, African spiny mice, Mongolian gerbils, and finches. So welcome, Dr. Kelly, and thank you for joining us today.
Thank you. It does sound like my lab has a bit of a circus going on.
Yeah, I was going to ask you, I mean, what is it like to, most labs you hear about, they specialize on one animal. What's it like to try to run a lab where you're dealing with so many different species and different needs? And
yeah, it's been interesting. In previous labs, I had been working with usually just one species at a time. Although with the birds, we had 5 different species of closely related finches, but they all had the exact same needs. So that was very, very easy. But with the rodents we have right now, they do all have their own specific needs. It can be a bit of a pain in the butt. The vet staff at Emory is actually incredibly helpful in making sure that they kind of each get their own dedicated spaces and working with us to make sure that their specific needs can be met. So it's actually more doable than one would think.
well, that's good to hear. And do you have to design like different special experimental apparatus for each species and like that? And
yeah, so the African spiny mice, they are relatively new to the study of social behavior and neuroscience. And when we first started testing them, we would put them in, kind of behavioral experiments that you would use for a regular rat or mouse or even a prairie vole. And the spiny mice, we found out that if they couldn't actually physically interact with each other, they would just be like, no, I don't really feel like doing this. If you're going to deny me the physical interaction of this social environment, then I'm not participating. So we had to just kind of redesign some of the experiments such that it's okay, just let them freely interact, or if you don't quite want them touching, then you had to drill bigger holes in a barrier so that they could actually get half of their head through. So it is kind of watching them behave and then tinkering the test or the apparatus to make it such that they'll behave in a way that at least somewhat mimics how they would behave in the wild.
Yeah, so that's kind of interesting. In a way, you can think of that as sort of a methodological hurdle, but in another way, it's already kind of a result that's telling you about the different nature of the sociality in these different species.
Yeah, absolutely. Yeah, the spiny mice are particularly social and pro-social, so a lot of positive social behaviors. They're also just really nonchalant little critters. So it's if you're working with a prairie vole, their number one goal in life is just to bite the experimenter and try to escape. But the spiny mice, if you're not careful, they will just run right up you. And they're just really bold, exploratory, kind of nonchalant about life. There's a field study showing that they actually don't respond to predator cues, which doesn't seem very smart, but it kind of fits in with their just yeah, we just go with the flow sort of behavioral phenotype. They're a fun species.
That is pretty cool. And if I understand, this is sort of the comparative logic, is you could start to try to understand why they have these sorts of differences. I don't know if that's sort of the avenue you're going down, or if there's other things that you're looking at.
Yeah, so with the spiny mice, they're so bizarrely social that kind of one thing that we like to think about is when you get these extreme animals, what happened in the brain, what happened over evolutionary time to allow them to become that way. But then we can try and compare them to a close relative. The easiest relative that we can get our hands on is the Mongolian gerbil. In fact, spiny mice aren't mice, they're very poorly named. They're more closely related to gerbils. They should be in the gerbil family, not the mouse family. But that way we can take a close relative like the Mongolian gerbil, who's not quite as social, and then we can kind of compare like what changed over time to allow the spiny mice to be just cooperative and so highly social, whereas the Mongolian gerbils are more hesitant and tentative in social situations and just generally a bit more territorial. So yeah, that's where that comparative approach comes into play.
Right. And you've got, I mean, I was looking at your lab website, you've got a lot of different tools that you use for making those comparisons. Like, you know, you just described some behavioral differences, but you also get into the genetics and the neuroanatomy. So, you know, what kinds of things are you looking at with the spiny mice?
Yeah, so we have primarily been focusing on the non-peptide system. So these are the vasopressin and oxytocin peptides that probably most people are familiar with. Oxytocin, which has been inappropriately dubbed the love hormone. I mean, it can also promote aggression, so it does a lot of things, as well as regulate basic physiology. But they're peptides that are really strongly evolutionarily conserved, and they're present in some form in bees and ants. They're in fish and lizards, frogs, they're in everybody, us included. And so what we're really interested in doing is seeing how flexible these peptides have been over time and how they can contribute to behavior. So with the spiny mice, nobody had ever looked at vasopressin or oxytocin in the brain. So one of the first things that we had to do was just validate antibodies that allow us to see these things and to see the neurons that produce these peptides. And to then see, okay, where are they? Are they in the same location of the brain as other animals? For the most part, yes, but there are some random populations that the spiny mice have that other animals don't have. So moving forward, we can then kind of go in and look at how these specific cell populations of oxytocin, vasopressin directly contribute to behavior. And if the weird neuronal populations present maybe contribute to the weird behavior that we see in them. So then, you know, we can do similar things in a variety of species as long as we can just confirm that we're looking at what we think we're looking at. In a new species, it's always tough because there are a lot of validation steps to make sure that you're actually labeling what you think you're labeling. But that's where genetics come in and sequencing, and we have wonderful collaborators, and the core facilities at Emory help us do that when there are things that we can't do in our own lab.
Okay, so you're using the genetics to help you make sure that you're labeling the neuronal systems that you think you're labeling. Did I get that correctly?
Yeah, for the most part. Yeah, and then we can go in and use technology like chemogenetics, which is using viral vectors to be able to go in and infect the brain and a specific cell type so that we can go in and we can tell the brain, turn off this population or turn on this population to see how that influences behavior.
Wow, so a viral brain lesion, huh?
Yeah, you know, it's kind of, I love the viral systems that have become available because it's straight out of a sci-fi book. That, you know, if you think about what do viruses do really well, they infect things. And so people far smarter than me went in and, you know, modified these viruses to be just delivery vectors, little shells that then people can kind of plug in the information that they want into this virus, and then you just inject it into the brain. It'll go where you want it to go and carry out the directions that you gave it. It's pretty incredible, and I'm very grateful for all those very intelligent bioengineers who can make the magic happen.
Yeah, that's pretty, I can imagine the science fiction book, although it seems like it might be a rather frightening science fiction.
Yeah.
You know, going back to like a step you were talking about, how these different peptides are kind of a shared toolkit that a lot of different species use. And so I'm wondering if that also gives you some leverage for translational sort of applications to humans, either in terms of basic science or health and well-being.
Yeah, one of the reasons that I really like the spiny mice is that they exhibit a social structure that's pretty similar to humans. When we think about the labs that do amazing research studying the neuroscience of bonding, they're using species that, like the prairie vole, that are socially monogamous. So you've got kind of these little tight nuclear families of male and a female partners, and then they have like a litter or two of offspring. But prairie voles, when they meet a stranger that they're not related to, who's not part of their family, they're really aggressive. And humans aren't really like that. We're not hanging out with our families and then go outside and beat the crap out of somebody that we see on the street. So prey voles are excellent for studying the mechanisms of parent-offspring relationships or bonds between mates. But when it comes to other types of social relationships, they're really not that great. And there are surprisingly few mammals that you can actually bring into a lab that exhibit a social structure like humans do. And the spiny mice are great for that. They have these big complex societies, and then they've got some, they'll still form like little groups within a larger group. But that allows us to be able to kind of mimic what human social relationships might look like when it's something outside of like a pair bond or parent-offspring relationship. And so at least behaviorally, we can have, a system that at least more closely resembles that of humans. And then we can go in and we can look at things like the peptides that are so strongly conserved that humans have vasopressin and oxytocin. And so instead of thinking, we should study maybe non-human primates because they're more closely related to a human, it might be more appropriate to think about the social structure that's similar, such that if you take a species like most rats, they don't exhibit paternal care, for example, that you probably don't want to be understanding the brain of a father using that rat model because they don't naturally exhibit this behavior. And so to find an animal that naturally exhibits this behavior, they may have independently evolved similar mechanisms that produce that behavior that then could be the same for humans. So it's kind of one of these arguments that instead of just looking for the closest relative, it might make more sense to find a species that exhibits the behavior that you care about. So we could find even from birds that just live lives that are more like humans, they may have evolved brain mechanisms similar to humans because that's what produces that same behavior. It doesn't always have to do with just a direct genetic relationship. So that's kind of one of the arguments for one, searching out the most appropriate species to ask your question in, and two, using that comparative approach. So, if we can identify what in birds or crazy rodents like spiny mice, even lizards and frogs and fish, if there's kind of these similar mechanisms that promote this kind of high sociality, and it's in all those species, then you can probably point to humans and say there's probably something really similar promoting those same behaviors in humans as well. So it's not necessarily the translational research in the disease-oriented sense, but more of how did our kind of whole behavioral phenotype evolve, and what might that look like?
Yeah, no, that makes a lot of sense. I think people have increasingly realized that sort of thing in human evolutionary studies within anthropology and so forth, that it's not always about how closely related you are, because there's so much convergence and the shared toolkit, this idea of like sort of deep homology. So we all have some of the same tools, and under certain circumstances, and what are those circumstances? That's very interesting. Which sort of leads me to think about, are there any sort of straightforward ecological sort of adaptive scenarios for these? Like, why are the spiny mice showing this kind of social structure versus the prairie voles?
Yeah, that's one of the fun things of getting into, what are the unique environmental demands that pushed spiny mice to be so social, that pushed humans to be so social. And that is something that we're slowly working on. Something that we have going on in the lab right now is looking at how the physical environment, like a lot like temperature, rainfall, how that can influence the non-peptides. So vasopressin is involved in maintaining cardiovascular tone. It's really important for blood pressure. It is critical for water retention properties in the body. And you can imagine that, let's say like a desert adapted species, they do really well without water for long periods of time. So presumably that ability to kind of modulate your physiology based on the physical environment, it's affecting the same system that also promotes social behavior. So we're kind of curious if, at least to a certain extent, the physical environment can almost place constraints on peptide systems that are important not just for physiology, but also social behavior. So we've got gerbils in the lab that are desert adapted, and we're kind of exposing them to different temperatures, to different kind of levels of water restriction. We've got pet brooders, which are supposed to be for incubating puppies, and then we've got gerbils in instead. But it is kind of one of these of just trying to get at that question of why did they evolve this particular phenotype? And so kind of starting out whether physical environment can have constraints on behavior.
Wow, that's really cool. Actually, it's sort of like an acceptation argument that might have been adapted, just for some water retention, but that then creates a circumstance in which it favors certain social, and because in human evolution, and we always think that everything about us is sort of special and purposeful and whatever. Everybody tries to explain the evolution of the social systems, the benefits of cooperation, and so forth. But of course, there have been major changes in our ecology at the same time, including the kinds of more arid habitats. So that's a really interesting idea. I'd love to talk to you more about that sometime and see what your results are. Anyway,
we'll find out.
That's pretty cool. That kind of leads me to a broader question I have, sort of theoretically, which I don't know if it's going to be a dud or interesting. But so you study social behavior. To what extent is there is that really like a different thing with its own dedicated neural systems and so forth, or is social behavior just behavior that happens in a social context?
Yeah, it's funny because in the psychology department, I'm surrounded by cognitive scientists who approach behavior and thought just in a very different way than I do. And my training is in biology, and I got my PhD in an evolutionary biology program. So it's been such a learning curve for me. Whenever I give a talk to my department, they always ask, but how do you know it's social? And at first my response is, well, how do you know it's not social? So kind of thinking about what is a social context at its kind of very basic level, at least to me, is any context when there's another animal around. They may not even be directly attending to them, but it's still a social context. And we often refer to the social brain and social neural circuitry. But no, I don't think that there's dedicated social neural circuitry, because most of the time when we do look at circuitry in the brain or chemicals or peptides that promote social behavior, they also do a whole lot of other things that aren't social at all. So it likely boils down to the connections they're making and multiple systems kind of working in concert in the brain to be able to constantly balance, considering the social context that they're in, but as well as everything else. An animal very rarely is just going to tune everything else out to just hang out with their buddy. Even we'll see in mating contexts, they'll be ready to mate because that's what we've evolved to do. But then if an intruder or a threat or something stressful happens, they can cut that off immediately. So they're clearly attending to other things going on. And I think it's, in a way, it's job security, but it's also incredibly frustrating that, we have this idea of there are networks in the brain, and there's a social behavior network, and then there's the memory network. And it's I don't know. It's nice theoretically to try and simplify it down into networks, but I just don't know that the brain's actually that simple.
Right. It kind of grabs what it needs to do the job.
Yeah. And it does seem that there are a lot of systems in place to be available. And to a certain extent, it seems like at a basal level, some are always on. So then it's just kind of maybe turn the dial up or down depending on the context.
Okay, well, that makes sense to me. So we've talked a lot about the rodents. So what are you doing with the finches?
Yeah, so we don't have any active finch work going on at the moment, but they with rodents, studying grouping social behaviors can be tough because there are so few rodents you can bring into the lab that do live in large groups. Spiny mice are great. We like working with the rodents because the technology to manipulate the brain is just fantastic. The technology to do that in birds is just slower to follow. We largely switched to rodent work because of the tech that's available. But birds are where it's at. I mean, there are so many different species that live in really large groups. They also communicate like humans do. Birds are primarily auditory and visual communicators. So it's a lot easier as a human to observe a bird and determine how they're interacting with each other, what's going on with rodents, because they primarily communicate through smells and olfaction. Sometimes you'll just see them like go up next to each other and you're just like, oh, I know I'm missing something between them. But with birds, you don't run into that problem. Something that we'd like to do moving forward is to work with fairy wrens, which are throughout Australia and New Zealand. And there is a paper that came out last week that was talking about how somebody went into the field and basically lived with fairy wrens for a year, which is awesome. And just, described their behavior. And they live in these really complex societies that are kind of at the level of humans and whales. And to be able to work with that species is kind of a dream that maybe we'll be able to do in the next few years. Just to be able to have, again, a different model for an animal that lives in these really complex, large groups, and to be able to start to understand the genetics, what in the brain promotes that preference to live in a large group, the flexibility to live in a large group, since you can't turn around and just beat everybody up. You've got to be nice enough some of the time, but also still be willing to engage in a little bit of aggression. So that's with birds, it'll be, again, kind of answering these questions about large group behaviors and understanding how societies are able to function and balance the good and the bad part of behavior.
Yeah, so they sound interesting, and we start talking about Australia and these complex social organizations. It makes me think of the reports of the cockatoos there with their tool use and their opening trash bins and all this kind of stuff. And the broader sort of idea that socially complex species are generally intelligent in lots of ways. And I don't know, is the aspect of social learning or animal culture or tool use or any of these things intersect with your interest in social behavior?
Yeah, it's something that one of my grad students is thinking about looking at in the Spiny Mice of, if you're living in a large group, do you remember everybody? Do you have to learn a lot about everybody and store that away in your memory somewhere? Or is it easier to not have to remember anything and just be ultimately flexible? So it's a hard question to answer in an animal that you can't just talk to. And ask, like, why are you doing that? Yeah, what did you learn from this? So it's a challenge, but it's such an excellent question of trying to figure out, are they learning? Are they rapid learners? Do they just not bother with learning a lot of information about other individuals, and instead they just go with the flow?
Cool. Well, so we've talked a lot about the work that you're doing and your interests. And I wonder, in your opinion, like what's the frontier? What's like the big stuff that you'd really like to get to that you're not there yet, or the next big question?
Yeah, I think a frontier that I'm ill-equipped to enter is looking at brain networks. So in our field, we have something called the social behavior network, which is a neural network in the brain. And it was conceived about 1999 with the idea that there are this group of about 6 brain regions that are really conserved. They are present across all vertebrate taxa, and they're all interconnected, and they're critical for all social behaviors. And a problem I have with that is that the idea, again, is nice, but the way in which we came up with this network is really biased. And I think this is where machine learning will come in and be extra helpful, is that instead of just having a human try to put together a network based on our limited knowledge, I mean, all you have to do is do a PubMed search, and there are just tens upon thousands of papers that we can't read all of them. And so to be able to have computers help us with that, to be able to identify all the brain regions or neurochemical types that may be involved in X behavior of interest, and to then use whole brain mapping and let it be more of a data-driven approach to identify this is a network that's involved all the time in this behavior, instead of kind of going in and cherry picking the few regions that we're familiar with. That requires graph theory and a lot of math that I just do not know. There are certainly some people at Penn and Harvard that are starting to do this, but they haven't really applied it to behavior. So I think that there are some neat collaborations that I would like to get involved with at some point, to kind of just really understand how, instead of looking at one or two brain regions and just vasopressin or just oxytocin, to try and get closer to that whole brain kind of approach to understanding how it is that we do what we do.
So, I mean, would this be similar to sort of the meta-analytical approach that people have taken to human neuroimaging data? Am I understanding that, or is this not?
Somewhat, yes, but with non-human animals, I mean, we can get just a lot deeper into the brain, and we can really get to the very specific cell types. So the types of data that we can gather from non-human animals is just, we can just get more because we can euthanize the animal at the end of the experiment, and kind of really get into depths of the brain that we're limited with humans. So it would be something that would get us more at the cell type level, and gene expression, specific areas of the brain, which we can't really do yet with humans. But it is using a lot of the stats and math that people in the fMRI world use, combining that with then the modeling of graph theory.
Wow, it does sound like a big project.
Yeah, it sounds like something that I have ideas about, but have no idea how to do.
Yeah, well, the machines will save us, right?
Yeah.
It's the artificial intelligence. Cool. Well, that's really cool. I guess those are mainly the questions that I had sort of jotted down. Was there anything else going on in your lab that you're particularly excited about that we haven't touched on? Any amusing anecdotes?
Yeah, we definitely do have the spiny mice will occasionally escape. So the Division of Animal Resources has gotten better at finding very specific like cage lids and stuff to keep them in, but for a while they had these like plastic lids that would just pop onto like their little mansion cages, and the spiny mice do excellent backflips. They're very acrobatic, and so we think that they probably just backflipped into the door and it popped off the cage, and then they went and had a grand old time. And so it is you get a call at strange hours of the day saying there's an empty cage that used to contain 5 mice, and so then we all have to go looking through the hallways because they can squeeze to very tiny spots, and they're usually like 8 feet high somewhere up on a rack inside a bag. The best was one that was just made it into the food bin, and it was just sitting in there just like chowing down. So they everybody on
made it to the food bin.
Yeah.
That's awesome. Well, I can imagine that would be quite startling to open your book bag or something.
Yeah, they're little now. They're very sweet looking.
At least they're not venomous or something,
Yeah, no. There is a venomous rodent out there, though, I think. In Australia, everything in Australia.
Yeah, of course. But it would have to be. But, yeah. Wow. So now I want to know about the evolution of spiny mouse backflipping behavior, but yeah. Say that's for another lab.
Yeah, I need somebody who studies like a motor output.
Yeah. Cool. Well, thank you very much. I really appreciate it. That's been very interesting. So.